Thank you very much, Matthew, and thank you very much for all the organizers to giving me the opportunity to present my research here. To bring everybody to the same page, I would like to just give a brief history about myself. I finished my PhD at UMass Medical School, and I primarily worked on the higher order genome architecture at the DNA level in the context of cellular differentiation as well as cancer. Then, of course, we know the nucleus does not only consist of RNA and proteins. There's the whole world of RNA. I don't have to tell this audience. So I switched to Harvard University and the Broad Institute and worked with Dr. John Rinn, worked on the RNA in shaping nuclear architecture. And to extend my postdoctoral training, in 2018, I joined Dr. Ben Blankov's laboratory at University of Toronto. And as you will be hearing today, I characterized the transcript and alternative splicing landscape of nuclear domains with a talented graduate student, Minkan Wu. When we think about the nucleus, it's a very crowded environment. Looking at the DNA level, the naked DNA is wrapped around the nucleosomes, which can be marked by different histone modifications. And then long-range interactions occur where promoters and their cis-regulatory elements, such as enhancers, are in close three-dimensional proximity. Then several of these interactions are clustered into topologically associated domains or chromatin compartments, which can be open or closed, heterochromatin or euchromatic. And many of these structures are heavily implicated in disease and differentiation. Ultimately, all of these structures make up the chromosome territories, where each chromosome inside the nucleus occupies its own distinct territory and show little intermingling with other chromosomes, which can vary across different cell states. Of course, on top of all these structures, the different molecular complexes with shared functions are selectively partitioned into different nuclear domains or condensates, as we recently call them. And there are many different flavors. To give a few examples, Cajal bodies are associated with small nuclear RNA processing. Nuclear speckles are associated with RNA splicing, RNA export. PML bodies are associated with viral host mechanisms, as well as the control of the genes that are associated with the cell cycle and DNA repair. Or the nucleolus, the most famous one, is associated with the ribosomal RNA expression. It is important to note that RNA can seed the formation of these subnuclear compartments via a mechanism called phase separation. By increasing the assembly and disassembly rate of the proteins and other components that ultimately mediate the nucleation of these nuclear bodies. And it does so by interacting with the multivalent domains or low complexity regions of proteins where RNA binding proteins or other transcription factors are enriched for, and catalyzes the interactions of multiple proteins within the same domain. Recently, Mitch Gutman's group from Caltech has shown that there are several non-coding RNAs that can help mediate the formation of distinct subnuclear structures, such as the nucleolus, small Cajal RNA hub, or the histone locus bodies. However, despite decades of research trying to characterize the molecular components of these nuclear bodies, where the protein composition is more readily characterized, the RNA composition has remained with only a few examples that we know. Some of these include the MALAT1 long non-coding RNA, which is heavily associated with progression of cancer and other diseases that are associated with the speckles. NEAT1 long non-coding RNA, a long link RNA that is required for the formation of paraspeckles, TAG1 link RNA with the polycomb body, and the ribosomal RNA with the nucleolus, alongside with a few more examples. The important thing is several components of these nuclear bodies are associated with disease. For example, as I just mentioned, the MALAT1 link RNA is associated not only with cancer and chemoresistance metastasis, but also a plethora of other diseases such as inflammation, angiogenesis, diabetes, or atherosclerosis. Similarly, the NEAT1 link RNA, which interacts with the SFPQ, PSPC1 mono or perispeckle proteins, apart from its role in cancer, is also associated with several non-cancerous diseases. Importantly, several drugs used in cancer treatments are partitioned into nuclear condensates. Work by Richard Young's group from MIT has shown that the widely used drug cisplatin, for instance, when labeled and the different proteins are labeled with GFP on the top, it interacts with mediator 1 in this case, as you can appreciate from the merged image, but not with other nuclear domain proteins, including fibrillarian or nucleophosmin-1 nucleolus, HB1 alpha, heterochromatin marker, BRD4, and SRSF2, a transcriptional activator as well as a splicing factor. Showing the specificity of this interaction, they mutated the aromatic residues of mediator 1, which resulted in the abrogation of the localization of this drug. And another drug called mitosectron also is interacting with a differential, shows differential localization with nucleolus as well as mediator 1 components, but not other factors, indicating that understanding the composition of these nuclear bodies will be important for several disease purposes apart from understanding the basic biology. So in this project, we sought to ask whether we can learn novel insights into nuclear processes by a systematic RNA mapping approach. And to do this, we use a recently developed method by Furkan Fazal in Alice Ting and Howard Cheng's laboratory, as well as from the Ignolia group, as well as from the Rin Alice Ting's group again, called ascorbyl-based peroxidates or APEX proximity labeling. APEX is an engineered APEX2 in this case, a protein or a tag, which can be genetically tagged to a protein of interest, in our case, nuclear body markers of interest, where they uniquely localize to distinct nuclear domains and expressed in the cells. And then the cells are briefly treated with biotin phenol as well as hydrogen peroxide, so that the APEX protein catalyzes the biotinylation of all DNA, RNA, and proteins within a 20 nanometer radius that are proximal to nuclear bodies of interest. And then this reaction is quenched, RNA is purified, pulled down by streptavidin beads, and then RNA-seq is performed. These studies have previously performed this technology to map the RNA composition of different organelle bound components, such as the mitochondria or the endoplasmic reticulum. However, it has not been done for subnuclear domains except the lamina. So in this project, we generated HEC293 flippant cell lines, which stably expressed APEX-seg and flag-tag nuclear speckle, Cajal body, PML body, nucleolus, SAM68 body, histone locus body, and nuclear periphery markers. To gauge for background interactions, we fused APEX2 with nuclear localization signal, flag, or GFP. Before doing, and all of these experiments were performed and co-led by Mikko Mu, a talented graduate student in the lab. Before doing all the high throughput experiments, we first wanted to validate whether APEX can specifically label subnuclear structures. To do this, we used a nuclear speckle localizing protein called SRSF7 that has been tagged with APEX as well as flag, and we performed immunofluorescence. On the top panel, where the cells have been APEX labeled by the addition of hydrogen peroxide, you can appreciate a robust biotinylation signal which is absent from the cells that have not been APEX labeled, but this construct is expressed, and which shows a high localization rate with the flag signal, as you can appreciate from the merged image. And furthermore, to see if these structures overlap with the endogenous nuclear speckles, we performed the immunofluorescence experiments in addition to including an endogenous SRM2 antibody. And from the line scan in the merged image, we observed a high localization between the biotinylation signal, the flag SRSF7 signal, as well as the endogenous SRM2 signal. Under the same experimental conditions, we pulled down RNA and performed GRT-PCR on select coding and non-coding RNA from cells that have been treated or not treated with hydrogen peroxide. As expected from literature evidence, we observed that the MALAT1 link RNA was the highest enriched compared to other RNAs, transcripts, as well as the non-labeled controls, indicating that APEX can specifically label these components. So we performed similar immunofluorescence experiments for multiple nuclear domains, such as the PML body marker, PML histone locus body marker, MPET, and the nuclear lamina, where we observed a nice overlap of the flag signal as well as with the biotinylation signal. Furthermore, in collaboration with Dr. Enclaude Ginira's laboratory at the Lannenfeld Institute in Toronto, we performed APEX mass spectrometry experiments to profile the protein interactome of these nuclear domains for select nuclear body markers, including the speckle markers, SRSF7, RMPS1, and SRSF1, PML body marker, SP100, and two nuclear periphery markers, lamin B1 and lamin A. And we identified a high correlation between the hits that have been pulled down with these markers for nuclear speckles, and they showed a higher correlation with compared to the hits, the correlation of the hits that are being pulled down by nuclear periphery markers or the PML body marker. In addition to this, proteins that have been pulled down by nuclear speckles were highly associated with spliceosomal complexes, as we observed gene ontology terms that include U2 type spliceosome complex or regulation of mRNA splicing, whereas the proteins that have been pulled down by the PML body marker SP100 were associated with the PML body sumotransferase activity and the double-strand DNA break remain. And finally, proteins that have been pulled down with nuclear periphery markers were associated with the nuclear envelope organization, further confirming the specificity of this method for non-organelle bound compartments. Encouraged by these results, we performed RNA sequencing on the RNAs that have been pulled down from these nuclear body, from the cells with these nuclear body marker APEX fusion constructs. And we also sequenced the input controls as well to assess the transcriptional and splicing changes that are that result from the increased expression of these proteins. First, when we look at the TSCNE clustering of APEX-seg replicates, we identified that the replicates that belong to the same compartment cluster together. And the different markers for the same nuclear compartment also cluster together. For example, SRSF7, SRSF1, and RMPS1 are relatively close together. SAM68, which is also involved in splicing, but is a relatively unknown nuclear body, is identical to splicing or in proximity to splicing speckle markers, and the PML body markers are in close proximity as well. In this heat map, I am showing you the transcript enrichment of around 12,000 transcripts that have been found to be significant over the NLS control in at least one of these markers. What you will notice is that many transcripts, each nuclear domain has a unique transcript profile that are partially shared with other nuclear domains. For instance, the speckle-associated transcripts are highly shared among the 3 nuclear speckle markers and not so much shared with the other nuclear domain markers. Lamin A has its own distinct cluster, which show partial overlaps with fibrillary nucleus marker or the histone locus body marker, as well as the PML and CAHAL body. SAM68, on the other hand, has its own distinct cluster that is separate from the nuclear speckle markers. In addition to that, PML body markers show a high correlation between the replicates, and fibrillary and MPET show some overlap. Interestingly, the CAHAL body markers REP53 and SMN2 does not really cluster together. And we identified that this is due to the partially cytoplasmic localization of SMN2, which can be both, according to the literature evidence, which can be both complex with REP53 in the nuclear CAHAL bodies, but also be on itself without complexing with Rep53 in the cell.
So, Rasim, I have a question here before we move on. So you just, I was going to ask actually about the overlap between the Hollow body markers, but then also the nuclear speckle markers. So in the paper, the numbers that you report for the transcripts are enriched with each of the three speckle markers is 21 to 28, 29%. So the fact that you're seeing this partial overlap, where do you think it comes from? Is it because of the regional organization of the speckle, where you might have a marker that's more specifically localized in one area of the speckle, or is it due to the binding preferences of your markers, or some other biochemical limitation that pertains to the resolution of APEX?
That's a great question, and the answer is probably a combination of all of the parameters that you've suggested. So, going to the first parameter, if you are thinking about the organization of the nuclear speckle and whether different transcripts are associated with different regions of the speckle, we would need to use many more nuclear speckle markers and perform APEX-seg and to see if they come up with a certain pattern or not. The second thing is the terminal, the protein terminal where APEX is fused. We identified that, for example, for SRSF1, if we take APEX 2 tag to the C terminus, we don't get a robust biotinylation, both by immunofluorescence as well as by QRT-PCR experiments. However, when we take it to the N terminal, then we get a successful tagging. And the same is true for other markers that we tried as well. Therefore, there is a biochemical, maybe a steric hindrance per se that can be going on, which is not identified and well characterized, but also the localization of these proteins in the nuclear speckles may affect it as well, though we don't have any experimental evidence yet.
All right. Thank you.
Yep. Yeah. So if we look at certain the golden example, MALAT1, for instance, and how they look in the genome tracks, it's primarily enriched in the nuclear speckle markers, but not with other nuclear domain markers or the controls. Another non-coding RNA called link 01126, which is unidentified with a much low expression transcript abundance levels, is also enriched in the nuclear speckle markers, but not with other nuclear domain markers or controls. Or another link RNA that is now associated with lamina. For the case of nuclear speckles, we identified around 150 additional link RNAs and over 800 coding RNAs that harbor similar enrichment to MALAT1. Therefore, this resource creates a big opportunity to identify regulatory and architectural RNAs. Looking at further examples, for example, the proto-oncogene KRAS is primarily associated with the SAM68 body, or the PDCD4, which is involved in programs cell death, is also associated with SAM64. A link RNA, which lies in an intronic region of the TBRG4 gene, the link RNA is called SLERT, which has been recently shown to be associated with the nucleolus, is found with the fibrillarium sample. And another example is decrin-1, which is involved in biolipid synthesis, is primarily involved in the two PML body markers, for instance. So what's different about the transcript that are associated with the different nuclear body markers? So we looked at the GC content and length and identified that for coding transcripts, the GC percentage was significantly higher compared to laminae, SAM68, or the average all transcripts. I'm comparing to laminae because they show the most extreme examples. And the speckle-associated transcripts were also shorter than the SAM68 or laminae-associated transcripts. For non-coding genes, even though we identified a higher GC content, interestingly, the transcript lengths were similar when compared to all transcripts or laminae pulled down transcripts. So Here, I would like to switch gears a little bit and talk about the alternative splicing landscape of these nuclear domains. As we all know, the alternative splicing results in different isoforms, which makes different isoforms of proteins as well. So that there can be alternative usage of promoters, poly-A sites, 5' and 3' splice sites, cassette exon or mutually exclusive exons, and retained introns, where the intronic regions are retained. And I will focus on the intron retention events. In this heat map, I am showing you the delta percent intron retained or delta PIR levels of transcripts that have been pulled down with these nuclear domain markers compared to the NLS pull down controls. So for those outside of the field, the red line indicates here that the transcripts had an intron that is enriched over the NLS control in this protein pull down. And a blue bar indicates that there was an exon-exon junction and the intron was removed. What you will immediately notice is that the speckle markers, as well as the nuclear periphery marker lamina, had a prominent enrichment of retained introns, whereas other markers showed trace amounts of intron retention events. And when we look at the overlap between these introns by an upset plot, where we identified over 5,000 introns in the speckle markers and the lamina, there was a very little overlap between the laminae-associated introns and the speckle-associated introns. And there was a significant overlap between each nuclear speckle marker as well. So for the rest of the talk, I would like to focus on the differences between these intron retention events. The nuclear speckle-associated introns, when we performed the gene ontology analysis, were primarily enriched for nuclear speckles, spliceosome, cell cycle, or translation initiation especially, because we identified a plethora of eukaryotic initiation factor genes with intron retention events that are associated with the speckles. On the other hand, the intron retention events associated with the lamina, nuclear periphery, belong to a completely different set of genes that were involved in non-coding RNA processing, chromosome organization, and microtubule organization, as well as GTPase activity, which is interesting because it's involved in nuclear import and export. So we identified that speckles and lamina are associated with different with transcripts having different functions. Next, we perform.
Rasim, before we move on, we have a question. So you mentioned this very briefly when you described the method. I just want to confirm with you that the RNA-seq was performed on ribodepleted RNA in all experiments, basically.
Yes, because we want to detect not only the processed transcripts but also the unprocessed transcripts, as well as non-coding RNAs that are not polyadenylated as well.
Right. Thank you for confirming that.
Next, we perform QRT experiments to validate some of these intron retention events. Therefore, we pulled down SRSF1 as well as laminae labeled transcripts and performed QRT-PCR on select speckle or laminae-associated introns, and observed a significant enrichment of speckle-associated intron recovery from SRSF1 pull-down samples in the labeled controls, and lamina-associated introns in the lamina apex pull-down samples. However, we did not detect any significant differences in the cells that have not been labeled with hydrogen peroxide treatment, confirming the specificity. How do these intron retention events look in the genome tracks? So even though there's considerably similar levels of exonic signal that's coming from all the samples, including controls, the speckle-associated introns showed a higher enrichment of these introns for the eukaryotic initiation factor 1 gene. A similar trend is true for the EIF3 gene as well, where we see increased intron retention here, as well as in this intron as well. And a similar trend is observed for the MTR or methionine synthase gene for the lamina sample. Next, we performed RNA fish experiments to validate the localization of these introns. So we selected MALAT1 target to probe for nuclear speckles, and we performed RNA-scope or RNA-fish using 3 speckle-associated introns, EIF1, DDX5, and EIF3G, and 1 lamina-associated intron, MTR. Just by looking at the images here, and the quantification is on the right, we observed that the speckle-associated introns were highly interior compared to the lamina-associated nuclear periphery-associated intron, as you can see from the quantification here. And the speckle-associated introns showed a significantly greater overlap with the MALAT1 signal, confirming their overlap with the nuclear speckles. Having all these data together, we were in a unique position to address novel questions. So I've been talking about different nuclear compartments, and the transcripts, 2 of them indicated, are here in black and orange, that are different. And in my introduction, I talked about the 2 feet at 2 meter or 6 feet DNA folded inside the nucleus. And there are the coding gene loci that are associated with it. Here, we asked, what is the spatial relationship between the nuclear body-associated transcripts with respect to their coding loci? And to address this question, we used a data set from Andrew Belmont's laboratory in Illinois called Tyramide Signal Amplification Sequencing, or TSA Seq method. This method is a proximity labeling method, but it is for DNA. They use the speckle-associated speckle protein called Son, which is a core component of nuclear speckles, and labeled all the DNA that is associated with the speckles. So when they sequence the DNA, the graph looks like this, where there are regions that are close to nuclear speckle, and there are regions that are far away. And by combining this genomic method with lots of DNA fish microscopy experiments, they were able to convert the genomic distances into actual microscopy micrometer distances, and came up with a cytological ruler, where they came up with 10 deciles, where the red decile, or number 10, was the most proximal to speckles, and number 1, the purple one, is the furthest away from the speckles. Using their data, we assessed whether coding regions of the transcripts that are associated with each nuclear domain marker belong to were either close to the speckles or farther away from the speckles. So we identified that speckle-associated transcripts were highly closer or in proximity to the speckles compared to the lamina-associated ones, as expected. In addition, we checked whether there's a trend for intron retention events as well. So by taking the 2 extreme deciles, number 1, which is the most furthest one, and number 10, we checked the percent of introns that are retained, more than 5%. And interestingly identified that for the nuclear speckle marker protein, SRSF1, if the coding region was close to the speckles, it showed a less splicing, where the intron was retained. And if the coding gene was away from the speckle, the intron was more spliced out, showing a less retention. And this trend was true for the other nuclear speckle markers, SRSF7 and RNPS1 as well. However, interestingly, this was not the trend for the lamina sample, where there wasn't any statistical significant difference between the retention levels. Furthermore, we in this study, in the Andrew Belmont study, they also performed TSA Seq for lamin C, lamin B1, Son, as well as SC35. And when we looked at the intron retention, the transcript enrichment, we identified that the genes that code for transcripts that are enriched for lamin A were significantly closer to the lamin C, lamin A TSA. So let me rephrase that. The transcripts that we found enriched by APEX-seg, close to the lamin A, their coding regions were also closer to the lamina sample. And this trend is the same for the lamin B sample as well. And however, the reverse pattern is observed where speckle-associated transcripts were close to either the SON or the SC35 proteins, more so than the lamina-associated transcripts or randomly selected transcripts, indicating an intricate spatial relationship between transcript localization as well as the coding gene localization in the three-dimensional nucleus. In my introduction, I've talked about genomic compartments, and that can be divided into A-type open or B-type closed, or euchromatic or heterochromatic compartments. And by microscopy, they can be interpreted as different nuclear condensates or domains. And by high-C heat maps, it can be shown as a checkerboard pattern. And a recent review by Job Decker's group suggested that, and by there are several other groups that suggested this as well, initially by Erez Lieberman's group, where the open type nuclear compartments are classified into two groups called A1 and A2. This is primarily based on their histone modifications. A1 is more associated with nuclear speckles because there's a study when SRM2 is knocked down, which is a core component of nuclear speckles. These regions are highly altered. And the A2 subcompartments are more associated with bromodomain proteins. On the other hand, the heterochromatic regions or the B-type compartments appear in three different flavors: B1, polycomp associated, B2, nucleolar associated domain or lamina associated domains associated, and B3, which is lamina associated domain or HB1 alpha. When we check the gene localization of the transcripts that are associated with the APEX-seg samples, we identified that the speckle-associated transcripts, their coding regions were highly associated with the A1 subcompartments, in comparison to lamina, where it was more enriched for the B2 and B3 compartments. Next, we wanted to assess whether there is a relationship by microscopy between the intron localization and their coding regions. Therefore, yes.
Yeah, before we go on, Rasim, I have a question, maybe a higher level question about what you just showed. So you show that, let's focus on introns are associated with speckles. So you show that these introns tend to be in genes that also localize close to speckles. So yes, it would be easy to speculate that because you have the speckle at the site of transcription, those speckles can mask nascent introns and prevent their co-transcriptional splicing.
Right? That is one idea. However, let me just bring up the data. We also performed poly-A RNA sequencing with the SRSF7 sample, and we identified that a majority of the introns that are also detected by the ribominus sample are also detected with the poly-A sample. So it is possible that most of these intron localization are not co-transcriptional; they can be post-transcriptional. And
So this brings me to the other possibility, which is whether you think that the presence of these introns can act as an anchor for speckles to recruit them at the locus of the gene.
Yes, that's a great hypothesis where I will explain some more evidence in a few slides that the retention of these introns might be interconnected with the formation of nuclear speckles.
Okay, very interesting. And maybe another follow-up question as to, so you've shown that there's a higher GC content in the transcripts that are in the speckles and also in the introns specifically, right?
That's also true, yes.
Right. Are there any other regulatory elements that you were able to identify that might explain why you have certain introns that are specifically associated with speckles?
I will be coming there in a few slides. We also cross-checked, intersected our data with the RNA encode data for ClipSig binding as well. And in a few slides, I will explain that we also identified several other speckle-associated RNA binding proteins that are bound to these introns as well.
Great, sounds good. Thanks.
Yeah. So just to recap this slide, we performed simultaneous RNA-DNA FISH where we visualized the coding region as well as the retained intron at the same time. And for the DDX5 intron, where we observed a lot of intron retention events that are spread throughout the nucleus and the coding regions labeled in white, we observed that of the RNA foci that we observed, intron foci that we observed, only 2% overlap with their coding region. And for EIF3 gene, where we observed less number of foci, only 15% of the EIF3G intron overlap with their coding region. And for the EIF1 gene, this rate was 41. So among all the thousands of intron retention events, we tested by microscopy only 3 of them, and we already found a wide range of overlap with their coding region. This indicates that, to us at least, that the speckle-associated introns may be both co-transcriptionally as well as post-transcriptionally regulated. However, in the case of nuclear lamina-associated MTR intron, the overlap was around over 80%, where we observed, first of all, a nice peripheral localization, both the DNA region as well as the intronic region, and there was a very high overlap between these two foci, indicating that.
Yeah, a quick question. So for the speckle associated introns, the FISH patterns are dramatically different, for example, between DDX5 and EIF41.
Yes.
Does that relate to transcript abundance or some other properties of the transcript?
Yes. So let me open that as well. So if we look at the transcript abundance of these introns, hopefully, okay. By looking at the genomic tracks, the intronic regions are here under about the CPM labels. And for DDX5, we observed around 15 counts per million reads, whereas for EIF1 and EIF3G, this range was from 2 to 3, basically. So
right.
that also
that makes sense.
Yes, and to add to your discussion, it is also possible that some of these foci that we detect by microscopy may represent intron lariats as well.
Very cool. Thanks.
Yep. So with this experiment, what it suggests us is that there is potentially differential co- and post-transcriptional regulation of these introns between the speckles and the lamina. So what's different about these introns? So first of all, the speckle-associated introns are shorter compared to lamina-associated ones or randomly selected ones. They are higher in GC content compared to the other two classes. If you look at the life cycle of an intron retention event, a retained intron, which may represent misprocessed transcripts, it can be exported to the cytoplasm and degraded by nonsense mediated decay. Or this retained intron can be spliced out. Then the mature mRNA can be exported to the cytoplasm and translated. And after translation, it can be put back to the nucleus and degraded by the exosome. However, in 2015, Philip Sharp's group, and now by Brown et al. In 2017, identified a subclass of retained introns called detained introns. These introns are stable inside the nucleus. They are not degraded by NMD or by other means. And upon external stimuli such as cell cycle or other external cues, these introns can be spliced out, and then the emanating RNA can be translated. Thus, this mechanism provides a readily available pool of transcripts that are post-transcriptionally processed, which can be used by the cell as a means of a rapid action. And when we looked at the overlap of these introns, the APEX-seg detected introns with detained introns, we identified a significant set of the speckle-associated introns are detained compared to lamina-associated or randomly selected ones. So, this is also really. Yes.
Yeah, we have a question on the chat. Can you maybe go back? I saw this quickly. I don't know if that relates to the question, so I'll just read the question. Can you tell us about the relative abundance of the transcripts that have retained introns versus the fully spliced transcripts?
That's a great question. We have not necessarily looked into this difference by detail, but I can tell you by inference that the transcripts with retained introns are closer to the speckles by TSA Seq method, and by TSA Seq method they showed that the regions that are closer to the speckles are highly expressed from the regions that are further away from the DNA regions from the further away from the speckles. So just by making that question, I would expect to see that the transcripts with retained introns are more highly transcribed compared to the more processed ones, but we have not directly looked at this in our data set.
All right, thank you.
So this is related to Matthew's question. In this APEX-seg study, we only used 3 nuclear speckle markers. However, we know that there are hundreds of different proteins that are enriched with nuclear speckles based on prior mass spectrometry data. And recently, several groups from the ENCODE consortium published 2 seminal papers where they analyzed more than 200 RNA protein bindings by ECLIPSE-seq, and they knocked down each of these RNA binding proteins or RBPs and followed the transcriptional and splicing changes by RNA-seq. And by microscopy, they also categorized the nuclear speckle-associated RNA binding proteins and also nuclear but non-nuclear speckle localizing RBPs. So by using all of this data and intersecting with the APEX-seg dataset, we identified that knockdown of speckle localized RBPs compared to nuclear but non-speckle localized RBPs, and also compared to lamina-associated or randomly selected introns, we observed that the speckle-associated introns are highly regulated by the knockdown of these speckle-associated other RBPs. And similarly, when we looked at the ECLIPSE-seq binding, we also observed a higher and significant enrichment of CLIP-seq peaks by speckle-associated RBPs on the speckle-associated introns, suggesting that many more proteins might contribute to the regulation of these or association with these introns.
Okay, so that answers a question that we had on the chat. Can you give us an idea of the number of RBPs that were associated with speckles that were knocked down in this experiment, and the number of lamina-associated RBPs?
Yes. So it's in the range of tens. Actually, I have it's also in our supplementary table, which we plotted like this. So
there you go. Yeah.
In this bar plot, you're looking at the log2 odds ratio of the different proteins. This is the binding CLIP-seq data that are either enriched in the speckle-associated introns or lamina-associated introns. And in this list, there are several proteins like HRMPA1 or SF3B4, for instance, that are canonical splicing proteins. And so it may be possible that these proteins are associated or contribute to the regulation of the speckle-associated introns, which we detect by SRSF1, SRSF7, and RNPS1. Right. I hope this answers the question.
Thank you.
All right. So moving forward, we identified several differences in the features of these introns, but how about their tissue-specific regulation? Therefore, we looked at different tissue-specific RNA-seq data sets and plotted the percentage of overlapping intron retention events that are above more than 20% retained, and identified that speckle-associated introns were highly retained in the pancreatic tissues, interestingly, and tissues associated with either blood tissue or the brain tissue. However, this is the top 20 tissue or cell types. If you go to the bottom of the list, interestingly, we observe mostly embryonic stem cell-like tissues where we did not observe any differential enrichment of these introns. This can be, excuse me, excuse me. This can be associated with the active transcriptional rate of these tissue types as well. Nuclear speckles are interesting, by the way. Okay, nuclear speckles are interesting because throughout the mitosis, they dissolve in early prophase and then reappear back in mid-telophase or early G1. And they follow the progression of the cell cycle in terms of assembly and disassembly. And indeed, consistent with this, prior study showing that knockdown of Son, which is a core component of nuclear speckles, as assessed by the SRSF1 staining here, leads to abrogation of or dissolution of nuclear speckles. And this is accompanied by a high mitotic index, where the cells basically cannot proceed with the cell cycle progression. The cell cycle progression is arrested. So when we looked at a RNA-seq data set for looking at the effects of Son knockdown, we identified that a higher percentage, significantly higher percentage of the introns that are regulated by Son overlap with the speckle-associated introns compared to lamina-associated or randomly selected ones. So having all these data together, we next wondered whether these introns are regulated across the cell cycle. Therefore, we used the RNA-seq data set from this paper, Dominguez et al., where they blocked the cell and then after releasing the cell, they performed RNA-seq experiments in 2 consecutive cell cycles from the same experiment. And analyzing this data, we identified that a significant fraction, close to 25 to 30%, of the speckle-associated introns were highly regulated throughout the cell cycle. And among those events, which are categorized into 7 different classes, one particular group, Group B in this case, which is orange, interestingly showed a reduction of retention levels in mitosis G1 transition and an increase in retention in G1S. And this was cyclic in the second cell cycle as well. Importantly, the transcripts inside this Group B introns were highly associated with mitotic cell cycle progression and included several important genes such as aurora kinase B or CDC like dual specificity kinase CLK1 or CLK4, which literature evidence have shown that they play a role in the assembly of nuclear speckles as well. So in summary, I've shown this one of the first surveys of nuclear domain-associated transcripts by a proximity ligation method for subnuclear compartments. And then I talked about distinct hubs of retained introns with differing length, GC content, detention levels, and gene ontology terms. And I talked about the intricate relationship between the coding DNA region localization and the transcript localization and how they are associated and related with each other. And then I talked about the different regulatory properties of these retained introns in different tissues and across the cell cycle. This indicates that the inclusion or retention of these introns are intricately related with the formation of the nuclear speckles, but more work is required to assess the mechanistic relationship. So I am currently on the job market for academic positions, and in my future laboratory or work, I aim to use look at the RNA localization in conjunction with the chromosomal folding of the nucleus to study several molecular pathways in several biological contexts. So with that, I would like to thank the Blanco Lab, especially Minkan Wu, where we co-led this project together, and he is looking for a postdoctoral position. So if he applies to your lab, do get him. And Claude Gingras lab from LTRI, and our collaborators, Philip Mas and Robert Wederit, and the Sequencing Center, and also my funding. And with that, I would like to thank you, and we'll be happy to take any questions.
Yeah, great work, Rasim. Beautiful presentation and beautiful work. Thank you very much. We have several questions on the chat. I'll probably start with one that's a follow-up on the questions about the relationship of between RBPs and retained introns. The question is, have you or has anybody tried to knock down the RBPs that are expected to bind those transcripts and to see if the localization is affected?
So based on the ENCODE data, some of the RNA binding proteins that are bound to these, significantly bound to these introns, or where these introns are regulated by the knockdown of these RBPs, they have the genomic data, but I don't think any localization assessment of these introns have been performed under the knockdown conditions of those RBPs.
Right, I'm not familiar with any such data either, but an interesting question. We have a technical question here. Why did you choose the specific speckle markers that you use, like SRSF1 and 7, instead of other known speckle markers like Son and ST35?
That's a great question, and this was based on a proximity biotinylation by mass spectrometry data from our collaborators that's not published. And based on that data, we identified that these three nuclear speckle proteins represent the variety of the proteins that are associated with the speckles, based on different profiling of different markers. And that was the main reason why we decided to use these three markers.
All right. Now I have a question that's it's both technical, but also it brings up a biological question. And the question is whether you have validated the speckle-associated transcripts in another cell line. And I think that's interesting because, technical validation, but also it speaks to how conserved are these possible programs of intron retention and localization in different systems, different tissues, or cell types?
Excellent question, and we have not performed RNA fish in using a different cell line, and it will be of great interest to repeat these experiments using different cell lines or tissue types even, and to see if the localization of these introns are conserved between the cell types, even the species, between mouse and human, for example, or are they, if they are not conserved, do they have any functional effect or any function in terms of cellular outcomes? That will be a great way to analyze it.
Absolutely. All right, we've got Forkan Fasal on the call, who was an important contributor to the development of APEX-seg. He's got questions for you. Any insights from APEX proteomics and correlation with APEX RNAs?
In terms of so you're asking about whether the proteins that we observe by APEX-seg, do we observe whether the transcripts are also enriched? We have not primarily looked into this. It will be interesting to look at it, yes. But we did compare APEX mass spectrometry data with BioID mass spectrometry data, and we observed a high correlation between the two methods. Depending on the different thresholds you choose, for bioinformatics, the rate changes varies a little bit, but we observed that many of the proteins that are detected by APEX mass spectrometry are also detected by the BioID, over 70%, to give a number. Yeah.
All right. Did you make any important changes from the original APEX-seg protocol?
So that's a great question. One primary change was we did very little tweaks to your original protocol, Furkan. And one of them was at the isolation step of the RNA, where we used the Ignolia method, where we just treated the streptavidin weeds with triazole and extracted the RNA from it instead of incubating in 55 for 2 hours, etc. And apart from that, the protocol was basically the same. One thing I should note, though, that different nuclear body markers, based on their sizes, for example, Cajal bodies are much smaller than nuclear speckles. We had to increase the number of cells that we used. And we don't have any conclusive evidence, but we think that the RNA that is pulled down depended on the actual size of the structure that you're working on.
All right. So you said you talked briefly about this in your introduction. There is some things are known about how RNA can contribute to the nucleation of membraneless organelles. Did you see any evidence, or do you have any feeling that these introns might be promoting phase separation, either for speckles or in other compartments?
That's a great question, and we don't have a mechanistic answer to that. Based on the nuclear speckle assembly and the differential inclusion of these introns during the cell cycle, we hypothesize that the retention of these introns, maybe in association with the transcriptional activity as well, is associated with the formation of nuclear speckles, because transcription also shuts down during mitosis mostly. So it could be possible that these introns might act as scaffolds for some of these nuclear domains, and some of these detained introns could be part of this regulatory mechanism. But it will be interesting to maybe knock out some of these introns that are promising, look promising, and then see the nuclear domain morphology. And some other ways would be if we, let's say, take an intron, nuclear speckle-associated intron, and put it in a lamina-associated transcripts, can we change the localization of this transcript within the nucleus? That will be an interesting avenue as well.
Right. All right. On another topic, did you look at N6A modifications in transcripts that are artificially located, I guess, or localized?
We did. It's not included in the paper, but we do see that nuclear speckle markers are somewhat enriched for the M6A modifications. Yes.
Oh, very cool. And maybe one last question before we go. So there is a great emphasis on retained intron in the paper. What about other types of alternative splicing events, and maybe in particular microexons, although it might not be the best system to look at those?
Microexons are primarily involved in the neuronal tissues. Therefore, we did not find. I mean, we did check whether microexons are differentially enriched, and we did not find a particular pattern. And we also looked at cassette exon and 3' and 5' alternative splice site usage events as well, and they are in the supplementary figures. And even though there are some introns that are enriched in certain nuclear domains or markers, there wasn't a striking pattern that's as striking as the intron retention events. So therefore, we did not pay too much attention on those events. Although I think it could be important to delve into those data sets as well.